In this video, we will discuss how to analyze the sequencing results of your 16S rRNA PCR product to aid in identifying your unknown bacterium. Before we begin, please ensure that you have downloaded the software to view your electropherogram sequence file. Eton Biosciences, the company that sequenced our PCR products, provides links to different electropherogram software packages. If you have an Apple computer, we suggest using the 4Peaks software package. And if you are using a Windows computer, we suggest using BioEdit. The link to the Eton Biosciences download page can be found on Moodle. Let's refresh ourselves on how we got to this point in our experiments. After setting up your cucumber fermentation jar, you performed a serial dilution viable plate count assay on an aliquot of pickle brine solution, plating on both MRS and VRBG agar plates. You then isolated one colony from one of your MRS dilution plates by T-streaking that colony onto a fresh sterile MRS plate to obtain a pure culture of that bacterium. From that T-streak plate, you selected one isolated colony to use in a polymerase chain reaction, where you amplified the 16S rRNA gene from the genome utilizing primers specific to the 16S rRNA gene. The product from your PCR was then sent to Eton Biosciences, where they sequenced your PCR product. It's important to note that the sequencing reaction is a different experiment than the PCR. The purpose of the PCR was to amplify one gene, specifically the 16S rRNA gene. The sequencing reaction then utilized that PCR product to assess the individual nucleotides that make up that specific gene. Remember, the 16S rRNA gene encodes for the 16S ribosomal RNA, which is an essential RNA molecule that is part of the ribosomal complex and required for the process of translation. In the image on the right is the secondary structure of the 16S ribosomal RNA. It is a single-stranded RNA that self-base pairs to form this complex secondary structure. We use this specific gene for two main reasons. First, all bacteria have a 16S ribosomal RNA gene. This gene is present on the chromosome of all known bacteria. Second, even though all bacteria have this gene, each bacterium's gene sequence is a little different. It's analogous to a human fingerprint. Fingerprints may look similar, but they all have slight differences providing a unique signature. This is very similar to the 16S ribosomal RNA gene. The 16S rRNA gene is approximately 1,500 nucleotides long, comprised of both conserved and variable regions. Conserved regions have the same or similar sequence among all bacteria. They are not unique enough to be used in identifying an unknown bacterium. Variable regions, however, have different sequences among different bacteria and can be used in identification. The sequencing experiment will sequence both conserved and variable regions of the 16S rRNA gene, but it is the differences in the variable regions that allow us to differentiate and ID an unknown bacterium. Your sequence and electropherogram files are available to download on Moodle in the 16S sequencing topic section. Remember that you will need to download the sequence viewer files, either 4Peaks or BioEdit, before being able to access and view these files. Let's first access your electropherogram file by clicking on the electropherogram file link on Moodle. Inside this folder, there are all the sequencing files from your section. Please reference the number highlighted in red on this screen to help locate your specific sample file. The sample number that is highlighted in red is the number of your PCR reaction tube from your previous lab session. Once you identify your sample, click the link and the file will automatically download. Next, you can download the sequence text file. The file name will be similar to that of the electropherogram file name. Locate your sample ID and click the link to download your file. Let's open your sequence text file first. This will most likely open automatically in a basic text program like Notepad. If your computer asks you what program you would like to use to open this file, please select a simple text viewing application like Notepad or TextEdit. When you open this file, you will see the sequence that Eton Biosciences recorded for your 16S PCR product. The question you need to ask is this sequence good? What portion of this sequence should I use in my analysis to help identify my unknown bacterium? The sequencing technology used by Eton Biosciences is called Sanger sequencing, or dideoxy termination sequencing. This type of sequencing usually produces around 600 to 800 accurate nucleotides, or base pairs of sequence. We only want to use good, accurate sequence in our analysis. So what portion of this sequence should we use for our analysis? To answer this question, we will need to view the electropherogram file. Please open your electropherogram file that you previously downloaded. This file should automatically open in the Sequence Viewer program that you downloaded from the Eton Biosciences website, either 4Peaks or BioEdit. Again, if your computer asks you what program you would like to use to open this file, select one of these two programs that you previously downloaded. For this demonstration, we'll be utilizing 4Peaks. BioEdit works in a very similar manner. When you open your file, you should see your electropherogram, which is an image of red, black, green, and blue peaks. This is a pictorial representation of your sequence that also provides information regarding how accurate the nucleotide base call is. Each of the peaks represents a nucleotide base that was sequenced from your PCR product. The color represents the specific nucleotide, where black equals guanine, red equals thymine, blue equals cytosine, and green equals adenine. We can use this electropherogram to assess the quality and accuracy of our sequence. Current Sanger sequencing technologies use different color-coded fluorescent bases. During the sequencing reaction, a computer will detect these different fluorescent signals as they are being sequenced and record the base that corresponds with that specific color signal. For example, if the computer sees a green signal, it will input an A in the sequence. The sequence that the computer assigned based off these fluorescent peaks can be seen at the top of your electropherogram file. This technology allows for rapid and efficient sequences of short DNA sequences, like our 16S rRNA gene PCR product. However, it's not 100% perfect. There are sometimes errors. The electropherogram allows us to see the fluorescent signals that were detected during the sequence reaction, and what base the computer inputted based on the signal it detected. Good accurate sequence will be assigned based off single, slim, tall, non-overlapping peaks. The image on your screen is of a good accurate sequence run. Poor inaccurate sequences are based off of broad, overlapping, non-single peaks. The image on your screen is an example of a poor inaccurate sequence. You will even notice that the computer software had a hard time assessing what base it should input based on this poor signal. There will be gaps, and sometimes you will see the letter N. A letter N in this sequence indicates that the computer was unable to assign a specific nucleotide at the position, and inputted an N for reference. When you look at your electropherogram, you will notice that you will have some poor sequence at the beginning and some poor sequence at the end. However, you should have a large portion of good, accurate sequence in the middle. Sanger sequencing typically provides about 500 to 800 bases of accurate, reliable sequence. Typically, a good sequence will start around base pair 20 to 50 and go until about position 550 to 800. Again, this is an average, and your sequence may vary a little from this example. You will want to assess your electropherogram and select the good sequence. You want to start as close to the beginning of the sequence as possible, and hopefully get around 500 to 700 bases of good, reliable sequence. The more accurate sequence you get during your sequencing reaction run, the more data you have during the analysis portion to help identify your unknown sample. Let's take a look at an example to help demonstrate how to select good sequence to use in the analysis step. Your electropharogram file is available to download on Moodle. Once you log into Moodle, proceed to the 16S sequencing section and click the link titled Electropharogram Files. Find the file for your specific sample and click the link to download your file. After your file downloads, open the file utilizing either 4Peaks or BioEdit. This demonstration will be utilizing 4Peaks. Once you open your file, you should see your electropharogram. You can increase the size of the program file to make it easier for viewing. Remember, the purpose of the electropharogram is to assess the quality of your sequence. We only want to select sequence represented by individual, narrow, non-overlapping peaks. In this example, the T at position 17 is a good starting point. Please note that your starting point might not be base pair 17. Remember, we want to start where there is single, non-overlapping peaks. The sequences at the beginning, positions 0 through 16, are not good quality. They are broad, overlapping peaks. And you will notice at the top the computer had a hard time determining what base to put based off of this fluorescent signal. Starting at position 17, we see nice individual non-overlapping narrow peaks, indicating accurate, reliable sequence. As we scroll, we continue to see good, reliable sequence until around base pair 570, when the peaks start to get broad and a little overlapping. If we select the sequence between base pair 570 and base pair 17, we will get 554 bases of sequence to utilize in our future analysis. Remember, we don't want to use the entire sequence. We only want to utilize accurate, reliable sequence. Selecting non-accurate sequence may interfere with the proper identification of your isolate in the preceding steps. Only select accurate and reliable sequences based on electropharogram results. Once you have your sequence highlighted, you can copy that selection. You can now utilize this selected sequence in the next part of our analysis. To analyze our sequence, we will be utilizing the Basic Local Alignment Search Tool, or BLAST for short, housed and maintained by the National Center for Biotechnology Information, part of the National Library of Medicine, which is one of the 27 institutes of the National Institutes of Health. You can access the BLAST website via the link on Moodle in the 16S sequencing section. BLAST is a computer algorithm program that compares sequence data to a curated database. As our sample sequence is a nucleotide sequence, we will be utilizing the nucleotide BLAST, or BLAST-N. BLAST also allows you to search by other sequence types, like a protein BLAST, which is called BLAST-P. This searches based off amino acid sequence. Click on the nucleotide BLAST icon to continue with your analysis. Paste your copied nucleotide sequence from your electropharogram into the query box at the top of the BLAST-N website. For this analysis, you do not need to change any of the preset search parameters. Just click the blue BLAST icon at the bottom of the screen. As of 2019, the database, called GenBank, contained 380 billion nucleotide bases from more than 215 million individual sequences. When you hit the BLAST button, your inputted sequence will be compared to a database of 380 billion individual nucleotides from 215 million different sequences, looking for a match. Imagine taking your sequence of about 550 letters, or bases, and searching through 215 million samples for a match. For a reference comparison, the NC State libraries have around 4.5 million books. When your search is complete, you will see this result on your screen. Remember, the BLAST program just searched 380 billion different bases and found a match to your specific sequence in under 2 minutes. At the top of your report, you will see the query type, which is DNA, as we use BLAST-N with a nucleotide query entry. It will also report the query length, and for this example, it was 554 nucleotides long, or 554 individual bases. When you scroll down, you will see a list of individual sequence samples from the database that matched your sample. Most likely, you will see many samples that have a 100% identity match, meaning that all 554 bases matched that sample in the database. This gives you very high confidence that your sample is very similar, if not identical, to that sample in the database. It's very important to note that in this analysis, we can only confidently assign a genus to our unknown sample. We cannot, however, assign a species level. In this example, we can confidently state that our sample is in the genus Lactococcus, but we cannot say with 100% confidence that it is Lactococcus lactis. The reason is that we only used approximately 1/3 of the total length of the 16S rRNA gene, about 550 bases out of a total of 1,500 total bases, the total length of the 16S rRNA gene. As we only used a portion of the total 16S rRNA gene sequence, we can only identify the genus. Remember that the 16S rRNA gene is composed of many different constant and variable regions, and we only assessed a portion of those in this analysis. If we wanted to identify our sample down to the species level, we would need to include the entire 16S rRNA gene sequence, or all 1,500 bases. This would require us to perform additional sequence experiments where we sequence the middle and end of our PCR product. This is beyond the scope of this lab. To demonstrate this point further, if we scroll down the list of matches, we eventually find a match to a different species of Lactococcus, Lactococcus delbrueckii, also with a 100% identity match. For this project, we are only interested in the genus level identification. In this example, we identified our sample as a bacterium in the genus Lactococcus. These bacteria are lactic acid bacteria that are homofermenters, meaning that they produce only lactic acid as the sole product of glucose fermentation. These results are logical and fit with our project, as the bacterium was isolated from a cucumber fermentation jar, where the sole carbon source was glucose. This concludes our video on utilizing sequencing and BLAST analysis to identify a bacterial isolate. Utilizing this demonstration video, go back and assess your specific sequence to help identify the bacterium you isolated from your individual pickle jar.